Okay. Good evening, everyone. Uh, Today we are going to talk about molecular marker, and probably this will be your last part. Uh, Before going to DNA marker, or we are going to call genetic marker, we will see what is molecular marker. Whenever we are talking about molecular markers can be any component. Any component means it can be carbohydrate, it can be proteins, it can be nucleic acid, it can be some fragments, or it can be some chemicals. Any molecules which we use as a marker means for identification, for identification of a tissue, for identification of a disorder, for identification of a disease. So that we are going to call it molecular marker. So if you see the definition of molecular marker is, it is a gene. When we are coming, because in our context, because we are going to further going to talk about DNA marker or genetic marker, so that's why I have written it. It is a gene or DNA. Otherwise, normally molecular marker means any case of component, any compound. It can be carbohydrate also, it can be proteins also, it can be non-biological source also. It's like some environmental factors, some pollutants, some toxic components. So molecular marker can be anything by which we are going to identify or we are going to detect certain components, yeah, certain disease or disorders. So molecular marker in our context is it is a gene or DNA fragment with a known location, because we are going to identify a disease or disorder or abnormality in a certain tissue. So that's why we should understand the location of that particular molecules or DNA on the chromosomes that can be used to identify individuals or a species or certain disease, means abnormalities you can call it. So in our topic, molecular marker is a gene or DNA sequence or DNA fragments whose exact location we are knowing it, and so that it will help us for the identification of a species, for the identification of a individual or certain specific things. It can be described as a variation means, like I before also we have seen that all the human beings, more than 99% DNA almost same. Less than 1% DNA are different, and that means 99 1% means maximum portion of our DNA are you can call it similar. Only some differences are there, and that different whatever differences are there, whatever variations we are having, which is caused because of mutations or alteration or their arrangements. Like if we are having certain, like this is my chromosome, some case of DNA which is non-protein coding DNA. So if that is at this place, and think about this is a repetitive DNA. If this repetitive DNA, in my case, it will be repeated two times. So one repetition here and second repetition here. So it is going to be repeated in my DNA two times. If it is going to be repeated in certain other individual five times or 10 times or n number of times, so the x means appearances or the characteristic feature of the person is going to be different. Okay, means what I'm going to tell you, these non-coding proteins, non-coding genes also, their variations which is caused because of mutations, not only point mutations can be anything, alteration in their genes, their locations, their repetitions, their uh, numbers. So that is going to called as molecular marker here, and that we are going to use as an identification symbol. Different types of molecular markers we are going to have. SNP can be one of the molecular markers. There are a number of molecular markers we are going to have, and these molecular markers are RAPD, RFLP, AFLP, SCAR, SSR, SLP, SNP. Number of molecular markers we are having. If you will see the name of these molecular markers, like RAPD, that is random amplified polymorphic DNA. If you will see the name of RFLP, that is restriction fragment length polymorphism. These two, RAPD and RFLP, we will see in the detail. Other than that one, we are having AFLP. AFLP means it is amplified fragment length polymorphism. Then we are having SNP, that is single nucleotide polymorphism. Then we are having SSLP, SSR, SCAR, number of other things we are having, OP. oligonucleotide polymorphism, many different types of molecular markers we are having. Like if you'll see in this example, this is the genetic sequence of certain individuals. So like AGAC, these I am going to concentrate on these 4 nucleotide sequences. If this 4 nucleotide sequences, if you'll see here, AGAC, this is going to be repeated. So in certain individuals, like individual number 1, if this AGAC is going to be repeated 6 times, so their appearances, their characteristic feature will be different. And the same AGAC is going to be repeated 16 times in another individuals, their characteristic feature will be different. Same AGAC is going to be repeated in third individual 10 times, so their characteristic feature will be different, their properties will be different, their appearances will be different. So on the basis of this repetition of the repeated DNA, which we are going to call it polymorphic DNA, so the characteristic features are going to vary. There, okay. Is it clear?
Yes, sir.
Fine. Now, what is the significance of these molecular markers? Where we are going to use it? As I have told you, these molecular markers are going to be used for different purposes. Some of the important purposes I have told, it's written here, like it is going to be used in different fields, like in forensic science, for the identification of criminal investigation, paternity identification, for studying different kind of disease genes. So for studying any kind of alteration, polymorphic analysis, identification of individuals, then species identifications, organism identification, phylogenetic tree analysis, markers assisted breeding, assessment of demographic bottleneck for natural and different kind of disease analysis. So these are some of the important uses of these molecular markers, or which we are going to call it genetic marker or DNA markers. These are some of the examples of commonly used molecular markers. Not all the molecular markers we are going to use very frequently. What we will see RFLP and RAPD in details. Rest, if time permit, then later on we will see it. So RFLP is restriction fragment length polymorphism. SSLP single sequence length polymorphism. AFLP is amplified fragment length polymorphism. RAPD, random amplification of polymorphic DNA or random amplified polymorphic DNA. VNTR, variable number tandem repeats. SSR, microsatellite polymorphism, or sometimes it is also called as SSR, that is simple sequence repeats. SNP, single nucleotide polymorphism. STR, short tandem repeats. SFP, single feature polymorphism. DART, diversity array technology, RAD markers, restriction site associated DNA markers. These are some of the common molecular markers or DNA markers. If you will see in this case, most of them are polymorphic DNA. They are going to be repeated in n number of times in different individuals. So repetition number is going to vary person to person, and that variation in the repetition number ultimately going to cause the characteristic feature changes. Clear?
Yes, sir.
Fine.
Yes, sir.
Fine. Now, if you see the type of molecular markers. So, molecular markers or genetic markers or DNA markers can be divided into 2 classes. Biochemical markers or molecular markers. Biochemical markers means it can be some of the biochemical components, like it can be proteins or it can be amino acids, which detects the variation in the gene product level, such as change in the proteins or amino acids. And when we are talking about the molecular markers, which detects the variation in the DNA level, like mutations, change in the locations, addition, duplications, inversion, deletions, insertion, means any kind of mutation in the DNA. So one kind of molecular genetic marker, we are having biochemical markers. These molecular biochemical markers also going to be altered, maybe because of the genes. Like if some genes are going to be overexpressed, some genes are going to be sub-expressed. So their protein expression level are going to be changed. If overexpressed, protein concentration will be very high. If it will be less expressed, the protein concentration will be very less. Or other, we are going to identify by the repetition, alteration, or changes, deletion, addition in the DNA level. So here RFLP, RAPD, normally we are going to see these molecular markers where we will see the change in the DNA components, DNA fragments. So that was the classification on the basis of the component. Markers can exhibit two modes of inheritance. One is called as dominant recessive, or second is called as codominant. Probably you might be knowing what is dominant, what is recessive. Whenever any characters we are having, like for every character, two alleles are there. One alleles normally we are getting from our father, another alleles we are getting from mother. So two alleles we are getting from both the parents, one alleles, right? If for a dominant character, even a single alleles are there, like you might have heard about Mendel's crosses. So if capital T and T also there, because in this case capital T means tallness. In this case only one tallness gene are there, allele is there, even though the offspring will be tall. So why this type of genetic components means the genetic constitution will be like capital T, small t, the plant will be tall, because this is dominant character. So dominant means whenever we are having only even a single allele, and if that characteristic feature will be the expressible character, that is called as dominant character. But for the recessive character, we require both the alleles. If we don't have both the alleles, like if you don't have both the T together, small t together, plant will not be dwarf. So plant to be dwarf, this we require both the alleles from both the parents. Then only plant will be dwarf. So this type of characteristic feature are called as recessive. Means for recessive alleles, we require two alleles together. Then only it will be expressible. And second one is codominance. Codominance means when both the characteristic feature will be expressed together. Like either means the characteristic feature which is coming from father also and mother also, both character will be expressed together. That is called as codominance. So some of the markers are dominant, some of the markers are recessive, or some of the markers are codominant. We will see which markers are recessive, which markers are dominant, and which are co-recessive dominant. Now, if the genetic pattern of homozygotes means where both the characters are together, like either it will be capital T, or a small t along with a small t. So this is called as homozygous. Homo means both same, similar type of gametes, similar type of genetic constitution from both the parents. So if the genetic pattern of homozygotes can be distinguished from that of the heterozygotes, then the marker is said to be codominant. Generally, codominant markers are more informative than the dominant markers. If you'll see the pattern or the information we are getting, so in whatever information we are getting from the dominant recessive character, or whatever information we are getting from codominant character, certainly codominant character gives us better information, more informations in the genetic analysis. These are some of the examples, some of the classification of molecular markers like SSR. You need to remember it. If you are going for write the examinations, you need to remember these markers like SSR, STR, RFLP. These are codominant markers. RAPD, AFLP, ISSR. These are dominant markers. Dominant means we require from both the means even if from single parent will have that character, it will be expressed. And codominant means if either it will be have the mother, or it will have father will have, character will be expressed. So that is called as codominant. Like in this case, you can see this P1 and P2 are the parents. P1 is parent number 1 and P2 is parent number 2. And this is offspring. If you will see in this case, this gene, this is the pattern, the genetic pattern from the gene, means you can call it gel electrophoresis pattern. So this gene which is present in the father, that is also going to be expressed into the offspring. So this is F1 generation, is the generation where this gene from the parent number 1 is going to be expressed, and the gene which mother is having or the second parent is having, that is also going to be expressed. Means offspring have all the character which is either parent, any of the parents have, and both the character are expressed together. So they are called as codominant, because the characteristic feature from this one also and characteristic feature from this one also. Characteristic feature here means DNA. So the DNA coming from parent number 1 as well as parent number 2 are expressed into the offspring together at the same time. So they are called as codominant. If you will see in case of second picture, where parent only if P1 is having one particular gene, so offspring have only one characteristic feature from the parent. The second characteristic feature from the parent 2 means they don't have any gene. So means it might be possible that offspring does not have any gene. If this gene would not have, means then you can call it, or it may be like that one, parent number 2 have this particular gene, but offspring does not have this gene, it is blank. So offspring F1 generation is going to have only one pattern, only one characteristic from one of the parent, either from parent number 1 or parent number 2. In this case, only one character coming to the offspring, that is from P1, not from P2. So they are called as dominant character, because this is dominant pattern, this is recessive pattern. So recessive pattern parents characteristic feature does not expressible there, does not visible there. So this type of pattern are called as dominant pattern, yeah, dominant markers. And when the characteristic feature on both the offsprings are expressible, they are called as codominant. Is it clear? What is codominant? What is
Yes, sir, clear.
fine. Now, these are some of the basic questions which has been asked in CSIR. Probably everyone might be knowing about the CSIR examinations. So if you'll see the questions, this is on the basis of some of the molecular markers only. Polymorphic DNA sequence are used for molecular identifications, and some of the polymorphic DNA sequence which has been used, that is STR, short tandem repeats, and their information has been given here. SNP, single nucleotide polymorphic, their analysis has been given there, are used as a polymorphic DNA. The below table summarizes the status of autosomal SNP, autosomal STR, mitochondrial SNP, and Y-linked STR. So you can see here autosomal, Y-linked, and mitochondrial for 4 individuals. So the individuals are A, B, C, and D, and these are the study which has been done in these 4 individuals. We have to identify, in this case, from these 4 A, B, C, D, 2 offspring are there and 2 parents are there. So you have to identify which of them 2 pairs are parents are there. Either A and D are the parent and B, C are the offspring, or AC parent and BD offspring, BC parent or AD offspring, or CD parent and A and B offspring. So this is the question. Question you have understand, right?
Yes, sir.
Okay. So can anyone tell me answer? No. Okay. Look at it. Normally when we are having the fertilization, so because 2 parents are there, so certainly one will be from father side, other one will be the offspring side. So one, if we are going to talk about, one will be the from father side for time being, this one, and one from mother side. So normally during fertilization, the fertilization will take place and zygotes will form. In the case of zygote, the zygote have mitochondria, like, and the mitochondria which is present in this zygote comes from father side or mother side?
Mother side.
Mother side. Okay. So time being, if I can assume that A and B are the father, yeah, mother, whatever you can call it. For time being, just I'm going to assume A and B are the parent. So if A have STR13 and B have STR13, so certainly when you are going to do the genetic analysis, because if A is the mother, so certainly offspring should have 13 number. 13 STR. If this is the mother, then also offspring should have 13. So certainly A and B are not the parent. So one pair you are going to omit, that A and B are not the parent. Clear? Otherwise, here also you could have 13 number, you could have 13 number. Is it clear? Mitochondria kahin se to aata? From somewhere the mitochondria will come to the offspring. If A and B are the parent, so C and D would be the offspring. So either from here or from here, mitochondria should come there. And if mitochondria is coming there, so mitochondrial DNA will also come there. And if mitochondrial DNA will come there, some number of STR should have to be there. But even a single STR is not there in the offspring. So certainly A and B are not the parent. Is it clear?
Yes, sir.
Okay. Now if you'll see, if you are going to do this analysis only, SNP analysis in the autosomes. So zygote have autosomes from sperm also, from the ovum also, from mother side also, father side also, right? Here we are going to see two types of SNP pattern in the autosomes. One is C, and second one is CG. Clear? दो ही तरह के पैटर्न है. Here you are having CC, here you have CC, and these two are CG. Clear?
Yes, sir.
Okay. If the father will be C and C type, and mother is also C and C type, then what will be the offspring? Certainly, C offspring will be C and C. Never chances are C and G will be there, because none of the father or mother have CG. So both father or mother have CC only. So only offspring can have only one possibility, that they will have CC. Clear? नहीं समझ में?
Yes, sir.
Everyone is getting it, right? If father is having C and C, if you are going to cross CC with CC, what will be the possibility? Is any chances that offspring will have CG? Because none of the parent is having G. So there are no chances that offspring will have CG type. So if parents will be CC and CC type, so offspring will be CC type only. Never it will get CG type. Now one by one, if you'll take, this is CC type parent, and this is CC type parent. So if I'm going to take this option A and D, so this is going to be crossed with this one. So A will cross with D. So offspring should have only CC type of pattern, right? होना चाहिए?
Yes, sir.
But in this case, if A and D are the parent, so offspring is having CG type. So certainly A and D are also not the parent. Clear?
Yes, sir. Clear.
Okay. Similarly, if you see, if B and C are the parent, where we are having CG and going to cross with CG, so offspring will never have CC type. Always offspring will be C and G type, because here also CG and here also CG. So only one possibility, CG type. So if B and C are the parent, then what will happen? Then offspring will have CG. If BC are the parent, then A and D are the offspring. So offspring never will be CC type. So certainly B and C are also not the parent. Clear? Is it clear?
Yes, sir.
Okay. See, on the basis of the mitochondrial SNP or mitochondrial gene analysis, if CD cannot be the parent, because from one parent mitochondrial gene to come. So if here also, if no mitochondrial DNA is there, no mitochondrial DNA is there, so CD cannot be the parent. Either A and C will be there, or B and D will be there. Means one with the mitochondrial DNA, one without mitochondrial DNA. So we have only two options. Either AC or CD. So if you'll see analyze this one, so you will get the answer that A and C are the parent. So
Yes, sir.
okay, and if you will just I will erase everything and then we'll show it. Now if you'll see here, I am telling you A and C are the parent. So see, A, this one and this one is the pattern. So I am going to cross CC with CG. So offspring can be of CC type also, can be of CG type also, because we are having both the type of options. So this also can be possible and this also can be possible as offspring. Clear? Similarly, if we'll see 1312 is going to be crossed with 1314. So 13 also possible, 14 also possible. Here all the organisms have similar type of thing, so you cannot compare on the basis of this SNP. So on the basis of this one and on the basis of this one, you can confirm that A and C, this one is the parent and other two are the offspring. Clear?
Yes, sir. Clear.
Similarly, if you go to this question, this is another question which has been asked in June 2018 in CSR only. This is also on the basis of, just now we have seen one graph. Like given below is the molecular profile of two parental lines, P1 and P2, and their diversity, that F1. Now if you'll say, it is, you have to tell that the marker that is represented in the above figure is most likely to be. Now you can say the offspring have two genetic fragment, two genetic component in the, you can call it the gene analysis when we are talking. So from parent number 1, this gene comes, or you can call it from parent 2 also this gene comes. From parent 1, this gene comes, but parent 2 have this gene, you are not able to see this gene into the offspring. Means the characteristic feature from only parent 1 is coming to the individual offspring. All the character which is present in the parent 2 is not going to expressed into the offspring, because if parent 2 of character will be expressed, then this gene also should be visible here, but this is not the case. It means when we have only characteristics from one of the parent is expressible in the offspring, we are going to call it dominant or recessive or codominance. Codominance means both the character from both the offspring, both the parent, right? Like you can see here. You'll see here, if all the characters from both the parents are expressible to the offspring, we are going to call it codominance. But when the characteristic feature of only one parent will be expressed, it is called as dominant character. So in this case, only characteristic feature from the P1 is visible. It means this type of characteristic features are the dominant character, dominant nature. So you have to find out which one is the pair are dominant only. You had seen the graph earlier. RFLP, SSR, or RAPD. SSR and RFLP are, you can see here, SSR codominant, RFLP codominant. So you can see here, RFLP codominant, SSR codominant, but this is dominant characteristic feature. So this is not possible. This is not possible. SSR is codominant, but RAPD is dominant character. So this is also not the correct answer. So answer is this pattern which is expressed here, that is dominant character, and out of this one only RAPD is dominant pair. So RAPD is the answer. Clear?
Yes, sir.
If I would have option like this one, this is I have created this one. So offspring have all the character from both the parent. See, both of this gene is there in the offspring. Like this is also here, this is also here. From parent 2, this is also here and this is also here. Then this would be a codominant type of character, not only dominant, because character coming from both the parent are expressed into the this generation. So then this is the example of codominance, and this is the example of dominance. Is it clear for everyone?
Yes, sir.
Yes, sir.
Fine. Now, this was the basic things of the you can call it molecular markers. Now, if we are going to start with RAPD, that is called as random amplification of polymorphic DNA. If we are going to see the name RAPD, random means anywhere, in any places, not at a specific place, right? Amplification means PCR, polymerization. Polymorphic DNA, those DNA which is polymorphic in DNA, polymorphic in nature, going to be amplified randomly, means from at any places. So this is called as random amplification of polymorphic DNA, RAPD. Commonly we are pronounced as rapid. This is a type of, it is a kind of, you can call it advanced version of, you can call it, or extension of PCR, because in this case later on we will see we are going to use polymerization chain reaction. So this is a type of PCR, but the segment of DNA that we are going to amplify here are random, not specific. In the common PCR, what we have used it, we are going to use a specific DNA and a specific primer we have used. So this primer will bind at the specific place of our DNA and then amplify it. So that is the difference. This is going to be used PCR, but not a specific primer. We are going to use random primer, which will bind at the random places and then amplify the DNA. So that's why it is called as random amplification of polymorphic DNA. Clear?
Yes, sir.
So this is the definition of a genetic marker, which already we have done it. Now if you'll see RAPD, so random amplification of polymorphic DNA, rapid, pronounced as rapid. It is a type of extension of PCR reaction, but the segment of DNA which is going to be amplified are random. It is different from the conventional PCR that it needs only one primer for amplification. Normal PCR, when we have done it, you are already knowing about the PCR. There we have used only one pair of primer. But in this RAPD, we are going to have very large number of short arbitrary primers, many number of primers. The size of primer are normally, we are not going to use very specific primer. We are not going to use very long primer. The primer length will be very specific. I will tell you what is the significance of primer length. The primer can be designed without any experimenter having any genetic information, because randomly it is going to bind at random places. So this technique was developed by two different laboratories at the almost same time. William et al. In 1990s and Wells and McClure in 1990, and called as rapid and AP-PCR. So William has called it rapid, and Wells and McClure has called it RAP-PCR, means arbitrary primed PCR. Arbitrary primed PCR means any random places PCR will initiate and start. What is the basic principle of this RAPD? In this case, the single short oligonucleotide primer we are going to use, primer length is very specific here, which binds to many number of places. Randomly it will bind to n number of places and use to amplify DNA very randomly. Like this is my DNA length, so I am going to use a primer. So this primer is going to bind here also, here also, here also, here also, randomly many places. So it will elongate this DNA many fragments, like it will amplify up to here, it will amplify this fragment up to here, this portion will be amplified here, this will be amplified here. So arbitrarily it is going to bind at a specific place and amplify the DNA in the specific location. This means that the amplified fragment generated by PCR depends on the length and size, both the primers and the target genome, as you have seen it in this figure. So this length of DNA is very small. This length of DNA is larger than that one, and this is the longest fragment of DNA. So the DNA length whatever the product we are getting after the PCR, it depends on two specific fragment. One is length of your primer, size of your primer, and the size of the target DNA. If you are having longer DNA, many places DNA will bind. If you are having a smaller DNA, only at a small number of places your primer will bind. So primer is going to bind at places that depends on the length of the DNA and length of the primer. The assumption is made that the given DNA sequence will occur in the genome on opposite DNA strands in opposite orientation within a distance that is randomly amplified. Amplified fragments are roughly 0.5 to 5 kb in size ranges, are separated by agrogel electrophoresis after the PCR. You are going to separate this one. And we are going to see how many different types of fragments we are getting it. Like, so how we are going to decide how much length primer we require? What is the size of primer we need? It will be too long or too small. For timing, just assume why primer we are taking roughly 10 to 13 nucleotide in this case. In case of RAPD, if we are going to select a primer length of 26 base pair. So the possibility or probability of binding of this length of primer, this is the length of primer, means you are having a primer length of 26 base pair. In any DNA, its probability is 1 in 4 to the power 26, because length is 26 and 4 is the number of nucleotide we are having: adenine, thymine, cytosine, guanine. So this DNA, this much bigger primer, can bind in a specific DNA at this particular means probability 1 place in 4 multiplied by 10 to the power 26 DNA. Okay, when you are going to this one is this probability, you are going to divide it, you will get 4 multiplied by 10 to the power 15. Okay, if you will multiply this one for 26 times. Now the average human length of DNA, if I will take the complete human genome sequence, their length is, they are having roughly 1 multiplied by 10 to the power 9 number of nucleotide. So this is human genome, which is of 1 multiplied by 10 to the power 9 nucleotides are there. But this is is a smaller length. If you will take the larger size of primer, it will bind one place out of this nucleotide. So because your DNA length, human DNA length, is very small, so in this human length, if you are going to take 26 base pair long primer, it will not bind even a single places. So 1 multiplied by 10 to the power 9, which is the length of the human genome, divided by this possible probability, this will come this much. This is the value. Means not at a single place primer will bind. So if your primer length will be too large, no primer will bind at any places. So this is the example. So DNA will be intact, no primer will be binding. If you are going to take a smaller, very small primer, then what will happen? Like if I'm going to select a 7 base pair primer length, then what will happen? The possibility probability is 1 multiplied by 4 to the power 7. If you are going to divide, calculate this one, you will get this value. Now, you know that human DNA length is 1 multiplied by 10 to the power 9. So 1 multiplied by 10 to the power 9 divided by this value, this probability. This gives you an answer of 6.25 multiplied by 10 to the power 4. Means primer will bind at 62,500 times places, different places. Think about, you have a tube, test tube or Eppendorf. In that one you are having 62,500 DNA fragments, and that 62,500 fragments, polymorphic fragments. How you will separate on a gel electrophoresis 62,000 fragments? Bands you cannot separate it. It cannot be visualized. Means if your primer length will be 7 base pair, you will get too many DNA fragments. If your primer length will be very small, large, no binding is there. If primer length is very small, too much binding is there. So then if we'll say, if we are going to take a primer length of 13 base pair long, then what will happen? Then this is the possibility, the probability. After calculation, you will get this value. And when we are going to divide this means a human genome divided by this probability, we are going to get 15. Means if this is the base pair length, primer length, then this will bind out in total human genome at 15 places. So on gel electrophoresis, 15 bands can be separated. You can get 15 different bands. So this is looking like that possible. Okay, so that's why we are roughly taking 10 to 13 base pair long primer length. So significance of primer length is that is, if your primer length will be too long, it will not bind at a specific places. If your primer length will be very small, it will bind at many places. But if your primer length is rough on an average 10 to 13 base pair long, it will bind at sufficient number so that can be visualized or can be analyzed on gel electrophoresis. Is it clear? What is the significance of primer length?
Yes.
Okay. Then what is the use of this primer length? Like I have told you, different organisms have these. These are going to be called polymorphic DNA because you are going to have a primer, and primer binds at a specific places and many places normally with the polymorphic DNA because multiple repetitions are there. Now, if you'll say this example, in this case, we are having 4 different dogs, and roughly for their skin color, some of the protein or some of the genes are responsible. Because these 3 dogs have the similar type of skin cares or the hair color, so certainly their characteristic, their genes are the same. But this dog's skin color is different. Means whatever characteristic feature is present, one character which is present here, minimum one gene which is present in this dog, that is certainly absent in these dogs. So this dog is going to have one extra or one additional character which is absent in these 3 dogs. So when you are going to take the DNA from these 4 dogs, so we will take the DNA from dog 1, 2, 3, and 4. Use a random primers, arbitrary primer of 10 to 13 nucleotide in all the 4 cases, and amplify it, and then do the gel electrophoresis. This is from dog A, dog B, dog C, and dog D. If you will say this type of patterns, means this gene present in all the 4 dogs. So this is common. This gene is also common in all the 4 cases. This 3 is present in all the 4 cases. This last is present in all the 4 cases. This DNA which is present in this dog is certainly absent in all other 3 dogs. Means the skin color or the brown color or the purple, whatever color this dog is having, for that skin color or the hair color, this gene is responsible. So this gene makes a difference. Yeah, this dog is different from other dogs because of this particular gene. This is one possibility. Other possibility might be that all 3 dogs, forget about this one. Forget about this one. This is not present. In these 3 dogs, this gene will be there. And in the 4th dog, this gene is absent. Means these 3 genes, if these 3 genes are present in these 3 dogs, means this gene is responsible for the black skin color or black hair color, which is absent in this one. Then this color will be, means whenever any kind of expressible characters are different, it is not only because of the certain proteins, there are certain additional genes are there, or certain additional genes are going to be removed. So how we are going to perform it? We are also going to use this RAPD for the identification also. So we are going to take the microbial samples from the different sites, different places, like 2 separate places we have taken. This is from the site A and this is from the site B. We are going to take the random primer, and same primer we are going to use in both the places. Here we are going to have the different type of bacteria, different type of microbial samples, microbes. Here we are having different types of microbes. We are going to use RAPD, random amplification of the polymorphic DNA, and then we are going to do the gel electrophoresis. So this is from this sample and this is from this sample. And once you are going to analyze their pattern, so you can say the genetic constitutions are different. Certain genetic, like this is the common gene which is present in both, and some of the characteristics which is different in this bacteria, which is different in this bacteria. So those, because those genes which is present in all the species everywhere, that is called as monomorphic band, because it is present in both the cases. But there are certain characteristic feature which is absent in one of the species, present in another species, that is called as polymorphic bands. Similarly, in this case also, this band is called as polymorphic band because it is present only one species. In other species, it is absent, and these are called as monomorphic bands. This is called as monomorphic bands. What is the advantages of this RAPD? And there are certain advantages, certain disadvantages. Advantages are because you are going to use a single primer and single polymorphic tubes, single time PCR you are doing, so it is little bit fast. You don't require any kind of sequence informations because we are going to use a random primer. We are going to use it. So randoms, no sequence information previously needed. Like in normal PCR, we require the sequence information so that primer can be designed. Here we are going to prepare a random primer. So that's why it is called as random amplification. In this case, we are not going to use any DNA probe. DNA probe we are going to use for hybridization. Later on, we will see. Arbitrary primer are easily purchased. No blotting or hybridization process is going to occur. A small amount of DNA is required because ultimately you are going to do the polymerization. So because of the PCR, a small amount of DNA is sufficient. So these are advantages of this RAPD. But there are certain disadvantages because the primers are going to bind randomly, not at a specific place. So you cannot reproduce the DNA same result every time. If you are going to use this time, this primer is going to bind here. In next time, it may bind here. So primers bind randomly at random places. So reproducibility or repeatability of the result is not possible. Because RAPD marker is a dominant marker, so you cannot get the information from both the parents. Mismatch between the primer and template may occur sometime. And there are certain other factors which is going to affect PCR, like some kind of enzymes, inhibitors, or some other things which affect the processes. And sometimes contaminants are also going to affect the process. So these are disadvantages of this RAPD. Is it clear for everyone?
Sir, how will
Yes,
we
sir.
improve the result then?
No, every time for the same sample, like human sample, you are going to take DNA from my body only and doing the RAPD. Every time you will get the different type of result. Repetition will not be there. Means DNA will bind at random places. So repetition, same result you will not get every time. Certainly, if you are going to take the DNA from my body and DNA from your body, you will see the different bands. But every time similar type of pattern you will not see. Clear?
Yes, sir.
Okay. Well, significance, where we are going to use it, we are going to use for species differentiation, like we have seen evolutionary genetics, population analysis we are doing, polymorphic gene analysis we are doing, genetic diversity among the species, within the species, we are going to use this one. There are different variations of RAPD. Like one is called as arbitrary primed polymerase chain reaction, APPCR, which we have seen initially. It was discovered by two different scientific groups at the same time. But now in this case, we are going to use a longer primer than RAPD. In RAPD, normally we are going to use 10 to 13 base pair long primer. In case of this APPCR, we are going to use slightly longer primer so that it will not amplify too much. Second one is DNA amplification fingerprinting, DAF, where we are going to use a smaller length of primer can be used so that many number of fragments we will get after the polymerization. So here we are going to get on an average 5 to 8 base pair long primer. And third one is multiple arbitrary amplicon profiling map. The collective term for the techniques use single arbitrary primers. Arbitrary means again longer primer we are going to use here. So these are different variations. But most commonly we are going to use RAPD itself, not their variations. Whenever you require a specific type of information, then only we are going to use a specific test, means profiling. Otherwise, normal common RAPD we are going to use it. Second one is called as RFLP, that is called as a restriction fragment length polymorphism. In this case also we are going to do the polymorphic analysis, polymorphic gene analysis, but here we don't have restriction. Initially we have seen RAPD, that is random amplification. Here we don't have random amplification. Here we are having a restriction fragment. Random amplification means you had a gene, primer will bind at random places and then do the amplification. So that is called a random amplification of polymorphic DNA. In this case, we are going to use the term restriction. Everyone is knowing restriction enzymes, restriction endonuclease. So certainly in this case, we are going to use DNA sample which is going to be fragmented by restriction endonuclease, and then we are going to analyze them. So in this case, restriction fragment length polymorphism, RFLP, commonly we are going to pronounce it RFLP. This is a technique that exploits the variation in homologous DNA sequence known as polymorphism for the distinguished differentiation between among the individuals in the population or in the species. This is a type of polymorphic that results from the variation in the DNA sequence recognized by restriction enzymes, restriction endonuclease especially. And in this case, we are going to use gel electrophoresis to visualize RFLP. Here, we are not going to do polymerase chain reaction. So certainly the amount of sample you need is very high. You cannot use very less amount of DNA. In case of RAPD, we can use a small amount of DNA because PCR was done. In this case, we are not going to do PCR, so we require higher amount of samples. What are the steps of this one, RFLP? First, we are going to extract the DNA. Then this DNA will be digested by restriction endonuclease. After digestions, you have DNA this is going to be DNA is going to be fragmented by restriction enzymes. Whatever fragment you are going to get it, that is going to be gel electrophoresis will be done. So gel electrophoresis of DNA fragment will be done. Once you have a gel electrophoresis where different bands you are going to get it, this gel we are going to transfer onto the nitrocellulose or southern blotting. We are going to perform it. We are going to do the southern blotting. So the transfer of DNA from the gel to the blotting membrane. Once the DNA will be transferred, we are going to hybridize them with the specific DNA probes. DNA probes means DNA probes are a small bionucleotide fragments which will be complementary to your DNA. So steps, if you'll remember, in this case we are digesting them with restriction enzymes. In RAPD we had not digested it. Here we are going to transfer onto the nitrocellulose membrane. In RAPD we didn't did the transfer. Here we are going to use the labeled probe. There we have not used labeled probe. Here we are going to visualize this probe with the autoradiography. There we have not done this process. So the basic difference between RFLP and RAPD is, in one case we are going to do polymerase chain reaction. In another case we are going to use restriction enzyme fragmentation. In one case we have only done electrophoresis. In another case we have done electrophoresis but transfer the DNA sample onto the nitrocellulose membrane. Then we have used DNA probes, and lastly autoradiography. Okay, so this is the example. Sorry, this is the DNA sample which is going to be fragmented. This is the normal human sample where this restriction enzymes is going to be digested at places. So in this case, it is going to digest here. It will digest here. It will digest here in normal human. In case of abnormal human, whenever we are having a digigic pattern, that time they are going to digest at random places. So this place, it is going to be mutated. Because it is going to be mutated at this place, so this restriction enzyme is it is not going to work here. Means previously when it was normal, then 1 fragment, 2 fragment, 3 fragment, 4 fragment we use to get it. Now after the mutation, after the change in the genes, we are going to get only 1 fragment, 2 fragment, and 3rd fragment. So the number of fragments what we are going to get it, that is going to be changed because of the mutation. So when you are going to digest these both the samples, normal and the digigic sample or abnormal sample, and then run the gel electrophoresis, you will get this type of pattern. Many genes are the common, but there are certain fragment which is present in normal but absent in digigic, or certain genes which is absent in digigic pattern, not present in normal. So when you are going to transfer this DNA, all the DNA will be transferred. This is also going to be transferred in this one. This is also going to be transferred here. This is transferred. This is transferred. So all the bands will be transferred here. But when you are going to use DNA leveled probes, so leveled probe will bind only those DNA which is different in case of normal or digigic pattern. So you will identify only 3 bands in this case. This is present in normal but absent in digigic. This is present in normal but absent in digigic. This is present in digigic but absent in normal. So you will see only 3 bands which is present in normal and digigic. And that genes DNA you can purify, you can take it, and then you can analyze for them. So in this case, we have not done polymorphic. We have just done the fragmentation, gel electrophoresis, transfer, and autoradiography. This is the steps. So the application of this RFLP is to determine the status of genetic disease such as cystic fibrosis or any genetic disease which is going to be caused because of mutations. Okay. To determine or confirm the source of DNA samples, such as in the paternity testing or criminal investigation. Just I will show you one of the example of criminal investigation or paternity testing. Like sometime you might have seen cases where 2 male is going to tell that I am the father of this child, or we are getting some sample from the crime site, and we are going to have many suspects, and we are going to match their DNA samples. So there we are going to use this RAPD. In genetic mapping or determine recombination rates that shows the genetic distance between the loci, or to identify the carrier of a disease causing mutations. There are certain disadvantages also, because in this case we are not doing PCR, so you require a more amount of samples, more amount of DNA you require. So RFLP procedure requires numerous steps. Many steps are there. In previous case, we had only one step, PCR step. In this case, you are having first restriction enzyme steps, then gel electrophoresis, then transfer, southern blotting, then probe usage of probes, and then autoradiography. So many steps are there. And if you are having many steps, so the possibility of doing mistakes are more. And possibility of doing mistake, it will certainly take a longer time also. In previous case, PCR, because you have used PCR, so DNA sample can be amplified in hours. RFLP also requires large amount of DNA sample because no PCR is going to be involved in this process in this case. Due to numerous reasons such as these, PCR techniques has largely replaced RFLP in most applications. So because of all these disadvantages, RFLP is not used too much, and it has been, but not absolutely zero. We are going to use at certain places. We are going to use at many places. Like for SNP analysis, in human genome projects, we have used RFLP only. Like see, this is the crime. This sample, this is some blood sample we have got, or some DNA sample we have got from the crime site. Okay, so we have done the RFLP from the DNA sample which we have got from the crime site. So this is the pattern from the crime site. This is the victim. Means for time being, this victim has been murdered. So this is the blood sample for this victim. So if you will see the blood sample which we have found from the criminal's crime site, certainly not matching exactly with the victims, because there are certain genes which is present in crime site's blood sample not matching with the victim, and there are certain gen DNA fragment which is not matching from victim to the crime site, like this one present here but absent in the victim. This is present in the victim but absent in the crime site. This is present in the crime site but absent in the victim. Certainly this one. So certainly the blood sample which we have got from the crime site is not from the victim. Then for that one we are having three different suspects. Suspects are suspect number 1, suspect number 2, suspect number 3. Now if you are going to match their blood sample or the DNA pattern, like this DNA, sorry, if you'll see this DNA, this is matching with this one, right?
Yes, sir.
This one matching with suspect number 1 also, suspect number 2 also. Then this one matching with 1, sorry, and 2. This is matching only with the 2. This one is matching 1, 2, and this one matching with 1 and 2. So can you tell me which one is the culprit? Who is the killer?
Number 2.
Number 2, because all these DNA means bands which is present at the blood sample or the certain sample at the crime site matches exactly with the suspect number 2. Clear? So by this one we are doing DNA fingerprinting for the finding of the criminals. Similar things we are doing for paternity testing also. Like say, this is the child DNA. When we are going to identify done the RFLP from the child's DNA sample, we have got this one. This is the blood sample or DNA analysis for the mother sample. In the child, if you'll see, and there are two person who is going to claim that he is the father. So you are going to match the DNA sample of the child with mother and father. Like say, this DNA is matching with father number 2. This is matching with father number 2. Because father is dominant, so all the genes which is present in the father will certainly be present in the child, but all the genes which is present in the mother will need not to be present in the child. That's why this gene DNA you are not going able to see in the child, because it is recessive. Then this third coming from this father, because this is coming from present in both the father. This is extra. This is matching with the father number 1. This is matching with the father number 2. So who will be the father? Who is the actual father? Where maximum matching we are having. In case of father number 1, 1 matching, 2 matching. In father number 2, 1, 2, 3, 4, 5. So which one is the father of the child?
2.
Number 2. So by this one we are normally, if you see the court cases or many case time, court gives the paternity testing analysis has to be done. So there we are going to do the paternity testing done by this way. Clear how we are doing DNA fingerprinting? So these are RFLP only. We are taking the DNA sample from mother, taking DNA sample from offspring, taking DNA sample from the suspects, and then doing the random restriction fragment, and then doing the analysis. Clear?
Yes, sir.
Yes, sir.
Now this is the difference between, there are many number of not only RFLP, RAPD, there are many number of genetic analysis we are doing. One is AFLP, amplified fragment length polymorphism. This we have not studied here. I have created a video. I have a video on my channel. You can, if you want, you can see it. The major difference you should know under RFLP and RAPD. RFLP, you have seen this one. RFLP is a co-dominant marker, whereas RAPD is co-dominant marker, but AFLP is a dominant marker. In RFLP, where we are going to do the restriction fragment length. This is restriction fragment because we are going to use restriction endonuclease. So here we require restriction enzymes. No PCR requires. RAPD, that is random amplification. So we are going to use PCR. No restriction enzymes is needed. In this case, because PCR we are not going to use, so you require large quantity of DNA. In this case, because PCR is going to be used, so a small quantity of DNA is needed. Because here we are going to use PCR, so we require a primer. Primer need designing. Here we don't require any primer. In this case, we require DNA probe. You remember, we require southern blotting. We require autoradiography here. In this case, we don't require DNA probes. We don't require southern blotting. We don't require any kind of autoradiography. And AFLP is certainly a combination of RFLP and RAPD. So in this case, we are going to have restriction also and polymorphic PCR also. So we are going to need both restriction enzymes as well as PCR. So this require primer also, and it requires restriction enzymes also. So it is AFLP is the combination of RFLP and RAPD. Is it clear?
Yes, sir.
So this is all about molecular marker, RFLP, RAPD, genetic markers, DNA markers, and their usage. If you have any question, any things, you can ask me. This slide I will send it to you. Any question
Yes, sir.
anyone is having? Any question?
No, sir.
No.